So Betty apologizes for not being here, but she will be watching the recording. Okay?
Good. Okay, shall I start the introduction then?
Please
Sure.
do.
Well, it's just a super pleasure to introduce Paul, one of my very earliest. You were my second graduate student, but conceivably the third. He actually applied to Cornell to arrive in 1982, and we accepted him, of course. And then he actually deferred it for a year so he could go and spend a year in Berlin studying architecture. So I don't know if at this time, Paul, you hadn't quite decided whether biology was your future or whether architecture was more interesting, but you certainly came back full of interesting stories about your year in Berlin and could talk to us endlessly about the architecture of Berlin, which was really cool. Just fantastic time having you in the lab, of course. You know, anybody that knows Paul knows that he loves to banter all the time, endlessly on almost every topic, from science to the next practical joke we're going to play on one of the people in the lab, and so on. And he impressed me among all my graduate students as probably the most independent of them all. At that time, we were just starting to do our studies of the effects of bath-applied monoamines, which I gave a talk on several months ago. And he quite correctly criticized his work as being unrealistic, because, you know, who bath applies stuff. So he did his thesis on identification of the neurons that provide serotonin, and that's the GPR story that I think sounds like you're going to start with that, briefly go through that, right, Paul? Which is a fantastic story. Many of you have followed up, and there's been a number of more papers on this, some papers even coming out right now, as our understanding the GPR gets more and more interesting. It was interesting because at that time, it's no longer true, unfortunately, Cornell required all graduate students to have to fulfill the requirements for a minor that was outside of their area. And Paul was interested in evolution, so he did a minor in evolution, which required that he take some courses, and he actually worked with my next door neighbor, Amy McCune, who's an evolutionary biologist, and just caught on fire. And I think the rest is history. This interest in evolution became central to everything that Paul's done since then. Nothing makes sense except in the light of evolution, I think, is what Paul wakes up to in the morning, goes to sleep at night saying. And the work has just been extraordinary because of that. I did a brief postdoc at Brandeis with Erwin Levitan. I don't know how much time you spent up in Eve's lab while you were there, Paul, but he dipped his toe into molecular pharmacology, looking at agonists of certain types of glutamate receptors in aplesia, and the molecular pharmacology part didn't stick, but the mollusc part stuck. And from that point on, all of his work's been on molluscs of various types. He went off to the University of Texas and worked with Bill Frost as a research assistant professor, where he made another huge discovery, which is also part of your title, the phenomenon of intrinsic neuromodulation of neuromodulatory neurons that are actually part of the network that they modulate. And that really opened up all sorts of new directions. Paul moved to Georgia State, where he worked his way up the ladder from assistant professor to Regents professor, which I think is the highest honor you can get there. Continue to do this beautiful comparative evolutionary based work, also with neuromodulation, a lot of work still on serotonin, on how networks evolve and how networks function to be multifunctional and so on. And you moved in 2017 then to UMass in Amherst, where you are the director of neuroscience, and continue to do gorgeous work, include, I don't know if you're going to talk about the Bergio work. You are. This is an incredible story where you're combining every possible modern technique available to understand completely the brain of a small mollusk. And it's just a fantastic story. So we're really in for a treat today. Two little personal details. One of the things that distinguished Paul as a graduate student was that he was a fantastic dancer. and his then girlfriend, soon to be wife and still wife, Sarah, would blow everybody out of the water whenever we had parties dancing. I hope you guys are still dancing, because it was a pleasure to watch and to try to copy you. And the other thing is that he can take that dancing onto the ski slope, and he's a fantastic skier. And for many, many years, we've gone together to the Winter Conference on Brain Research and talked on science on the chairlift, and then skied down with great, great pleasure. So Paul's a man of many, many talents, many interests, and we're in for a real treat for the science today.
Well, thank you, Ron. That was such a generous introduction. And I hope it lives up to it. So if you have questions of clarification, please feel free to interrupt me and ask that. I'd like to have the interactions, but I've got a lot. This is the first time I've ever presented like from alpha to omega, everything from A to Z. And I have the unpublished stuff at the end. But there's slides in here, and making a deep dive, I wanted to talk to the stomatogastric community, because like you guys would be the only ones that are going to appreciate some of the minutia in here. So I did some deep dives into some work that I've actually never spoken about. So this is my journey from GPR to DSI to SI1. Starts with the crab, but ends up with Bergia, as Ron said. And there's a little rat in the way too, so not all mollusk. So there are themes that kept arising in my work, as Ron mentioned, identified neurons, neural circuits, connectomics, neuromodulation, diversity of actions, extrinsic and intrinsic neuromodulation, individual and species differences in circuits, and evolution of neural circuits and behavior. And a large part of my career is just plain old serendipity. Things happen by chance, coincidence, fate, fortune, and yields amazing opportunities. And the first piece of serendipity was actually the fact that I ended up going to Cornell and meeting Ron. This is Ron actually giving me advice when I was a very young graduate student, but this is his son. But it was just pure chance that, because Ron had just started there. He had come from Ed Kravitz's lab at Harvard, hadn't published on the stomatogastric yet. So the only way that I knew about it was visiting him. And the only reason I visited him was because my sister was going to Syracuse. And so I sort of invited myself for an interview, saying I was visiting my sister, which I wasn't, but that's a separate issue. And the whole basis for my thesis then took off from this paper that was published by second year in graduate school. It was, I think this, Ron, is this your first stomatogastric paper? Right, with Bob Flam, who was another student, and Scott Hooper was Eve's student, and then Barb Beltz and Judith Eisen. They actually identified the GPR cells, but they weren't sure that they were the source of all of the serotonin in the stomatogastric ganglion, and they weren't present. They couldn't find them in Homaris, and they couldn't find it, and there was no serotonin staining in Panularis. But there was, in looking back at this paper, I just went and looked back at that last night, and there was this cryptic acknowledgement, and I want to know the answer to this, Ron. Special thanks to an anonymous colleague who cheerfully transported the HPLC samples from Boston to Ithaca against all odds. Is this like a spy movie or something? What was going on?
I think it was a friend of mine who had a friend in Boston, a girlfriend in Boston or something. I can't remember the details, but it was just a friend.
And why did they need to remain anonymous, though? Is this like contraband?
Yeah, I mean, you don't want people to know where you get your sources of your HPLC material. That's secret stuff,
Oh, maybe they were stolen from Ed's lab? Oh, that's on tape. So the other part of the serendipity was actually discovering the GPR cells. So in Homaris, so nobody had found them before. This is actually from crab, because I couldn't find a picture from Homaris right away, the clawed lobster. But I did something stupid. I was doing electrophysiology and looking at the effects of serotonin on the pyloric neurons. And then I took that exact same prep that I just dumped 10 to the minus fourth molar serotonin on, and I ran it through immunohistochemistry against serotonin. And if I had thought for 2 seconds about that, I would have said, well, that's stupid. Everything's going to stain now because it's been soaking in serotonin. But it turned out that it was perfect for making these cells pop out. because they take up serotonin and the cell bodies normally don't stain very well. So it was that stupid mistake that took me 2 months to figure out what I had done to get it to work right that led to my thesis. Just serendipity. This is a beautiful drawing of the system by Wendy Sustoff-Pyries, who was another grad student at the time. But it shows the position of the two GPR cells in the crab, their projections into the stomatogastric ganglion, and then their continuation on into both commissure ganglia. There are species differences in the GPR cells. There's 4 of them in Homarus, and there's 2 in crab. The 4 cells co-innervate the muscles, whereas in crab, they're segregated to 2 different muscles. So it was an interesting evolutionary progression that appeared right early on in my graduate career, that I could see that there were differences in species and how these cells were acting. And then Eve's lab went on to find that the cells also were immunoreactive to a neuropeptide, allostatin. And then she graciously put on the lines from the back. She was very reticent to put the lines because all the descending input, but then I got to sneak in the back door. One of the first physiological, very robust electrophysiological effect of the GPR cells is to induce plateau potentials in the DG motor neuron in the stomatogastric ganglion. So this is showing if you give a train of spikes in GPR, you get these EPSPs that summate, and then they produce a prolonged plateau potential. If you just give a current pulse, there's no plateau potential. But if you stimulate and get the plateau potential, you can terminate it early with a brief downward current pulse, a hyperpolarization, brief hyperpolarization. And that the EPSPs don't have to trigger it, but you can trigger it with current pulses. And this is a transient effect that goes away after a while. So this was a very robust effect, and it was the mechanisms were then looked on later on by Ole Keen before he got to his Nobel speech. But he is now doing all sorts of work in the spinal cord, and he started as a postdoc in Ron's lab looking at the 2 different conductances in GG that underlie the production of the plateau potentials, the hyperpolarization activated inward current and the reduction of a transient outward current. But there's actually a 3rd conductance. So this one cell is being modulated in 3 different conductances. And this is Bing Zhang, who was a grad student after me. He showed that there was a calcium conductance that's also modulated by the DSIs. I'm sorry, skipping to the future, by the GPRs and serotonin. And he used a cage calcium here to initiate the plateau potential. We also found that the GPRs modulate the pyloric rhythm, so not just the DG, which is in the gastric mill rhythm. It increases the burst rate or initiates bursting in PD and causes LP to be excited and depolarize more. And Bing also showed that there was a variety of different serotonin receptor subtypes that are mediating different effects on different neurons. This is part of the theme that I've continued on in other nudibranchs as well, that serotonin has many different effects on different cells, and that they are mediated by different types of receptors, and that also has repercussions for the evolution of behavior. The other thing I learned as a grad student was how to shuck a crab. And this is important, turned out, for the behavior, because when you shuck a crab, that's the foregut right there that gets left behind, sorry, down here. And so the muscles of the gastric mill are visible when in the shell that's left over. And so I was able to then record the activity of the gastric mill system in a semi-intact preparation that was stuck onto the top of the carapace. It was upside down. And this appeared in the first paper. And I showed that there was rhythmic GPR activity that was timed to the movement of the gastric mill. But if you cut the LVN and the muscles are no longer moving, the cells kept bursting. This got me really nervous. I wasn't sure what to make of this, because is this just an artifact? Is this injury? I didn't know. You can isolate the nerve and see bursting that had nothing to do with the movement of the gastric mill. But then 10 years later, John Birmingham in Eve's lab validated this result, which was a real relief for me. They published this paper that showed that the bursting was actually quite important for how the information was coded, how the stretch information was coded. It was really, and then Mike Nussbaum has made an industry out of the GPR cells and figuring out how they're, and Don Blitz also, in figuring out how they relate to the larger circuitry in the somatogastric nervous system. So what was thought of as sensory input is actually quite intrinsic to how the system is working. So there were several lessons, I think, that were learned from GPR. One is that the sensory neurons can evoke neuromodulatory actions. This was quite novel at the time. The other is that neuromodulation involves a complicated mix of neurons, receptors, and conductances, and that there are species differences in the anatomy and physiology of identified neurons, and that the properties and release dynamics of modulatory neurons cause their effects to differ from bath-applied substances. So you can't really get some of these effects by just bath applying serotonin, and you get this really odd looking rhythm when you do. And that extrinsic neuromodulation is not so extrinsic. It can actually be intrinsic, depending on how you draw your lines around the circuitry. So then moving on from the stomatogastric ganglion, I'm going to do a postdoc at Boston University looking at neuromodulation of aplesia feeding, Claude, but you may wonder about those publications because this actually never happened. The guy I was going to work with, Mark Kirk, after I got an NIH postdoctoral fellowship, told me he took a job in Missouri. So I was going to start a GRASS fellowship and work on aplesia and look at the feeding system, but I had to scrounge around for something to do. But serendipity had it that CK Govind was there. CK was the loveliest person ever. He was a kind soul, and he did beautiful work. He was an electron microscopist. Unfortunately, he died too young, as electron microscopists are apt to do. And he has this work that I want to just put out here for people because it's unfinished work, unfinished life work that he didn't have the tools to complete. He was looking at what causes the claw asymmetry in lobsters. So lobsters always have a crusher claw and a cutter claw, but if you raise them with nothing to touch, then they get two cutter claws. And if you exercise just one of the claws, it will turn into the crusher claw. So it's an activity-dependent change in phenotype that is always unilateral. You can't get two crusher claws. And so there must be a mechanism that changes gene regulation, but inhibits the other side, and it's activity dependent. And that project is just sitting there waiting for somebody who has molecular techniques to look at the gene regulatory networks that are underlying it. But I didn't work on that with him. Instead, I followed up on an observation that I had made in the stomatogastric nervous system that the GM8 and GM9 muscles are both innervated by the MG motor neuron, but they have different dynamics of contraction when MG is activated. So if you activate MG just tonically, the EPSP, the EJPs in the GM8 muscle facilitate, whereas the EJPs in the GM9 muscle depress. And this leads to a gradual closing of the GM8 or contraction of the GM8 muscle and an immediate contraction and then a relaxing of the GM9 muscle, all in response to a single motor neuron. And CK did these beautiful EM images, and we show that there's differences in the neuromuscular junctions of these two muscles and in their quantal release properties that underlie this difference in the contraction rates. That summer, when I was at Woods Hole working with CK, I met Peter Getting, and Peter Getting worked on Tritonia, and I wish Eve was here because she'd tell you the story about how they were at a party and locked themselves in the bathroom to discuss how circuits can have different configurations. And this was the beginning of both of their thinking about multi-purpose neural circuits. And Peter wrote this review paper talking about the Tritonia circuit with the DSIs and how they get configured into a reflex withdrawal mode and a central pattern generator mode. Turns out the details of this are not quite right because he didn't know about the neuromodulation. Now, this was not my first introduction to Tritonia, and this would not be what caused me to then work on it. I had actually met Dennis Willis at my very first neuroscience meeting when it was in Boston, I believe. And he's the initiator of the work on Tritonia. This is from a Scientific American paper in the late 60s or early 70s, where he mapped out the neurons in the Tritonia brain and looked at their behavioral effects, what each of these individual neurons are doing. He was so gracious. I went up to his poster. I really remember this. I was a first-year graduate student. I went up to his poster, and standing in front of his poster was Eric Kandel, Al Silverstein, and 3 other people, big shot people. And they're all looking at his poster, and I'm standing there. I was a first-year graduate student. He turns to me, looks at my name tag, he goes, "So Paul, do you have any questions?" With all these other guys standing there. I was really impressed with that. He was such a welcoming person. But that's not why I went to work on Tritonia either. So here's, Ron was saying we skied together. So here's Erwin Levitan, who was my postdoc advisor, and Ron, and this was at a winter conference on brain research. So I got to ski with my 2 first mentors. But my time with Erwin actually wasn't very productive. So yes, I did a postdoc at Brandeis. It wasn't with Eve, it was with Erwin, and I got 1 paper out of it. This actually, there's been mistake. People think that I maybe did a postdoc with Eve because I was at Brandeis and I did do stomatogastric, but I didn't. In fact, when I gave a talk there recently, Chris Miller after talking with Chris, he brings me up to Eve's office and says, "Here's your boy." And Eve and I both kind of looked at each other and I was like, not really. So the reason that I work on Tritonia is because my wife got a job in Texas. So her postdoc was incredibly successful. She had papers in Science and Nature about the rewiring of cortex from visual to auditory. One of the reviewers had called it the most significant experiments on sensory perception since Aristotle. That's literally what the reviewer said. And so we moved to Houston, and this is where my next bit of serendipity came about, because I had heard Bill Frost talk about Tritonia at the Society for Neuroscience meeting after meeting Peter Getting, and Bill was in Houston. So I asked Bill if I could do a second postdoc with him, but he didn't have any money for me. So Jack Byrne, who was chair of the department, I think is still chair of the department, said I could be appointed research faculty contingent upon getting an NIH grant. So I wrote an NIH grant based on Bill's preliminary data and moved to Houston before we found out if it was funded. And that eventually led to a paper in Nature. And I also want to thank Eve, because Eve is actually partially responsible for me getting this paper in Nature because it was rejected. And Eve said, and I told Eve about that, and she said, every paper gets rejected. You have to just argue with them because the reviews were good. And so I went back and argued with them and we got it in. So if she hadn't said that to me, I wouldn't have a paper in Nature. And they published the picture of Tritonia upside down. So it's like, that's how I get it. But anyway, this actually, I should point out, this is Bill. We were at Friday Harbor, so this is on a collecting trip to get Tritonia, and here's our area where we do dissections and the rig and all. No computers in sight. So this is the Tritonia swim circuit, if you're not familiar with it. So Tritonia exhibits a dorsal ventral swimming pattern, and you can record from the neurons in the central pattern generator, C2, DSI, and VSI, the dorsal swim interneurons and the ventral swim interneurons. And the behavior lasts about a minute. The swimming pattern lasts about a minute. The dorsal swim interneurons cause the body to flex dorsally, and the ventral swim interneurons cause the body to flex ventrally. And it goes dorsal, ventral, and so forth. So unlike the somatogastric ganglion, here's the behavior that it produces. It's very simple. It's just an up and down movement. But like the stomatogastric ganglion, it exhibits temperature sensitivity. So this is the C2 bursts at 5 degrees, 10 degrees, and 15 degrees. And you can see that it shortens as the temperature goes up, but the phasing stays exactly the same. So this is very similar to what's been observed in the stomatogastric nervous system, but it's buried on figure 11 in this paper that nobody's read. So you're all excused for not knowing that. So this is what the brain of a Tritonia looks like. It's significantly bigger than a stomatogastric ganglion. Some of the neurons are bigger than the stomatogastric ganglion. So that's a neuron that's 200 microns in diameter. That neuron is C2, which is right there. The dorsal swim interneurons are right there, but it's hard to see if you haven't been trained, and also hard to see in this particular photomicrograph. But if you stain with an antibody against serotonin, then they pop right out. So they're a cluster of 3 neurons that's part of a larger cluster of 5 neurons. That's the metacerebral cell, which Claude has worked on. That's found in almost all gastropods. C2, of course, is not serotonergic, and then there's various other serotonergic neurons around the brain. So this is the pathway that's been worked out from Dennis Willows, Peter Getting, Bill Frost, and myself. We have the sensory neurons going to a trigger neuron, going to dorsal rampant neuron, to the central pattern generator, and then to efferent neurons that then go to the musculature. The finding that Bill had made that then we got into Nature, and interestingly, this was Katz, Getting, and Frost, and Peter Getting didn't really know who I was, but we're co-authors together because I had just met him a month. So that summer at Woods Hole when I met him, a month later, he had a brain aneurysm and was unable to form long-term memories after that. So it ended his scientific career. But Bill just told me that he found out that Peter's son is doing really well. So that's good news. He had a hard time, but then he's recovered and now he's doing very well. So the DSIs increase the strength of C2 synapses. And so here, this is the early work where you stimulate C2 to fire action potentials. You get an EPSP, giant EPSPs in a follower cell, and stimulate a DSI, and those EPSPs get even bigger. Evan Hill, who was a postdoc in my lab, showed that this was due to calcium increasing the increase in presynaptic calcium in C2. So this is showing a calcium image of the terminals of C2. Here C2 was stimulated to fire action potentials. This is the calcium signal in its neurite. This is now DSI stimulation, and this is the depolarization that's caused by DSI synapsing on C2, but it's not causing any change in the calcium in the terminals of C2. But if you stimulate C2 with DSI, so following DSI, here's C2 stimulation, that calcium signal goes up. So it's a contingent change in the calcium dynamics, contingent upon C2 firing action potentials. And the evidence that we have is that it's coming from intracellular stores, not calcium channels. The DSIs, so the DSIs, we call this intrinsic neuromodulation, and I did not coin that term. Claude actually published that term previously with respect to aplysia. I was ignorant of that at the time, though, and subsequently figured that out. So the DSIs have a neuromodulatory effect on another CPG neuron. So that means that while this circuit is working, DSI is changing the strength of C2 synapses. So it's not a static circuit. You can't talk about the strength of the synapse because the strength of the synapse here is different than the strength of the synapse when the DSIs are firing. And it also increases the strength of synapses made by the other CPG neuron, VSI. This is work by Akira Sakurai, who is a long-term collaborator. He's now a research scientist. He's been a research scientist in my lab at Georgia State and continues to work at Georgia State as a research scientist. Akira showed that if you stimulate a VSI, you get the nice EPSC. He did voltage clamps, so he always did more elegant experiments than I ever did. If you then stimulate a DSI here, 10 hertz, 5 seconds, and wait 3 seconds later, that EPSC is bigger. So it's another example of a presynaptic enhancement of synaptic strength. And we worked out a model for this, and Bob Callan-Jagaman was a postdoc in my lab. And I wish Eve was here because this story relates to her also. He's wearing a t-shirt, I love Eric Kandel, because he is a real practical joker. And he has impersonated Kandel several times. And one of those times was by printing out a name tag at the Society for Neuroscience meeting with Eric's name on it. And I went up to him and I said, "What are you doing?" So he pulled that off, and underneath it was Eve's name. And so he had Eve Martyr's name tag at the SFN meeting, and he had figured out how to hack the system, and it was a security breach. And Eve was president at the time, so I told her about that, and they fixed that security breach. But he was good at finding flaws like that in systems. But anyway, we modeled the system and found out that what DSI is doing is it's increasing the fraction of readily releasable vesicles every time DSI fires an action potential, and that's what's causing the enhancement of the synapse. But Akira found that it didn't just enhance VSI synapses, it could also decrease the VSI synapse. So this was really a quandary. If you stimulate DSI and wait 3 seconds before you stimulate VSI, the synapse got stronger. But if you wait 25 seconds, the synapse got weaker. And then Akira realized that it's not because of DSI, it's because of VSI. So VSI exhibits post-tetanic or homosynaptic potentiation. So if you just give a train of action potentials, the strength of that synapse stays elevated for over 10 minutes. If you then stimulate a DSI, the strength goes back down. So in other words, what DSI is doing, the dorsal swim interneuron is doing, it's releasing serotonin and it's depotentiating the synapse. If you stimulate a second time, it has absolutely no effect because it's already depotentiated. So the effect of DSI is dependent upon the state of the VSI synapse. If VSI is potentiated, DSI will depotentiate it. And that works with a 25-second delay. So here is what I just showed you, that you give a tetanus, you stimulate VSI every 30 seconds, and you depotentiate that synapse from the potentiated state. But if you did the same experiment in an unpotentiated system, there's no effect. So DSI releases serotonin, and there's absolutely no effect on VSI synapses if the system's not in a potentiated state and you wait 20 and there's 25 seconds between the two. But if there's 5 seconds between the two, then it potentiates it, which is what I showed you before. And if it's potentiated already, then you get this initial potentiation followed by depotentiation. So there's 4 different effects that DSI can have on VSI depending on the state of the system and the delay between DSI and VSI spikes. This is not mimicked by bath application because you lose all of that when you bath apply serotonin. We also tried to see what would happen if we just upped the serotonin concentration by treating it with the precursor for serotonin, 5-hydroxytryptophan. And what we found is that, yeah, it massively enhances the DSI action. So here's, again, this is one of those things I've never spoken about before, but only because this crowd would kind of get it. Here's a DSI stimulus. C2 is being stimulated periodically. You see the EPSPs evoked by DSI into the follower cell, and also a little tiny EPSP in C2. And then you see the increase in synaptic strength there, that one is, you can go 1 to 2. 2 is much bigger than 1. And then if you treat it with the precursor, you load the DSIs with serotonin, and now look at this whopping effect it has on C2. It has this massive effect on the postsynaptic cell. It's just like huge, way out of proportion. And now it goes from being this big to being this big. It's just massive modulation, but also massive synaptic actions. It shows that it's due to serotonin, but it also is just totally out of proportion to what the cell normally does. This is actually work by Dave Fickbaum, who started as an undergraduate in Ron's lab and then did a postdoc with me. I should have pointed that out before. So this is the normal swim motor pattern, and Dave used a carbon fiber electrode to see if he can record the serotonin signal using cyclic voltammetry. And there's really nothing to see. But if you treat it with 5-HTP, you get this large serotonin signal. So there's this massive release of serotonin during the swim motor pattern. But then the paradoxical thing is that the DSIs are not firing. So you get this massive increase. Here's the DSIs, what they normally look like. The whole motor pattern is kind of wonky because the C2s just keep going. And yet the DSIs are hardly firing at all. Part of the problem is that what 5-HTP does is it causes other cells to start releasing serotonin. So other cells take up 5-HTP, and these cells are never there. These cells are never there if you don't treat with 5-HTP. So now you're getting 5-HTP, you're getting serotonin in cells that are not normally serotonergic. So when you stimulate a body wall nerve to initiate a swim, lots of cells are releasing serotonin, and that sets the system off. And one of the things that it does is it feeds back inhibition onto the DSIs. This led us to say, well, people are taking 5-HTP as a nutraceutical. Does that mean that if you take 5-HTP, you're causing non-serotonergic neurons that have amino acid decarboxylase to convert 5-HTP into 5-HT? It's the same enzyme that converts L-DOPA into dopamine. So we looked in the substantia nigra of rats, And this was the only collaboration I've ever had scientifically with my wife, because she had the IACUC. I never wanted to fill out the animal care and use. But anyway, these are serotonin staining in the substantia nigra. Here's tyrosine hydroxylase staining. So this is a dopaminergic neuron, and it's staining for serotonin because we fed the rat the precursor for serotonin. So that means that this has physiological implications for mammals. So that's about DSI. It modulates T2 synaptic strength and excitability. Its actions are mediated by intracellular calcium. The DSI modulation of DSI synaptic strength is both state and timing dependent. Bath application is not the same as stimulating the cell, and precursor treatment can cause abnormal physiology. I can pause here if there are any questions, because I just went through that really fast and I've got more. But if anybody has any questions they want to unmute and ask, I can stop sharing too. I was going to tell the story next about individual differences between cells, but I want to give people a chance to ask. Did the baby want to ask anything, Millie? Okay. All right, I'll go on then. Farzan, I'm gonna tell you that you're not keeping up your end of the bargain of asking the hard questions unless you're just saving them. All right, so we found out that not all tritonia are created equal. And this is again work of Akira Sakurai. If you look at the connection from C2 to VSI, which I haven't told you about yet, if you stimulate C2, you get this biphasic synaptic action. There's initial depolarization followed by an after hyperpolarization. And if you look at another animal, sometimes that second hyperpolarization, the inhibitory component, is weaker. So here's two different animals, two different synaptic strengths, but their motor patterns are just about equivalent. We couldn't tell the difference looking at it ahead of time as to whether one was going to have strong inhibition or weak inhibition. But if you block the commissure that connects the two pedal ganglia, so this is the cells, I didn't mention, the C2 and VSI both go through the pedal commissure, and they actually synapse in both pedal ganglia. But if you block that commissure, then this animal, actually the swim motor pattern was impaired, and this animal was fine. And we found out that it was correlated with the strength of that inhibition. So the stronger that inhibition, the more the cycle period was changed. Okay, so the -5 corresponds to the size of that hyperpolarization, and the percentage is how many swim cycles was it decreased by. So Akira used the dynamic clamp to purposefully alter the strength of that synapse. So you can put electrodes in C2 and VSI, and then inject additional current when C2 is firing to cause the VSI synapse to be more inhibitory. And when he did that with the commissure intact, which is when we normally don't see any individual differences, so here's no dynamic clamp, and here's the same prep with the dynamic clamp to increase the inhibition, and there's no change in the number of cycles. So this is the number of cycles under control conditions, and this is the number of cycles with the dynamic clamp causing this large inhibition. And this is current injected to the soma, so the inhibition is visible from the recording because it's not happening out in the neuropil, it's happening right there in the soma. Then he did the same thing after blocking the commissure with TTX. So this is the time in which having lots of inhibition would make the animal susceptible to having that lesion. Here's the commissure block, but this animal has weak inhibition, so it's just fine. You block the commissure and it's exactly the same as it was before. But if you add the inhibition, now it's impaired. So having the inhibition and the commissure blocked caused the preps to produce fewer cycles than under control conditions. So the individual variability was varied. It wasn't visible under normal conditions. It's only when a lesion occurred that suddenly that individual variability made a difference. And Akira showed this also in whole animals, that he could cut the commissure and some animals would swim and some animals wouldn't swim. And they also recover from this. So here are two animals. They both had 5 or 6 cycle swims. cut the commissure, and they're both impaired in this case. So presumably they both had strong inhibition. Then wait a while, and this one recovered pretty well, and this one didn't. So it turned out that the difference in the recovery was due to recruitment of additional synapses, which I didn't show here. So additional neurons that we never figured out. There's other neurons that are coming in and being recruited during the recovery period. And this is the beginning of the chink in the identified neuron simple circuit problem that, assuming that everything is these small neurons, the large neurons that we know, and ignoring all the other neurons that we don't know. I'll get back to that. So Tritonia and Pleurobenchia both swim with dorsal ventral body flexions, but they evolved that independently. Pleurobenchia is not even a nudibranch. So there's a lot more species that I'm not showing here, but the most recent common ancestor of Tritonia and Pleurobenchia did not swim with dorsal ventral body flexions because the ancestral condition of nudibranchs was not dorsal ventral. Tritonia is one of the weird nudibranchs that swims that way. So that means that most likely they evolved this behavior independently or convergent evolution. Farzan, you're unmuted, so that means you're going to ask me a tough question. No, he's just back.
No.
Pleurobenchia has the same homologous neurons as Tritonia. So Tritonia said the DSIs, there's a cluster of 3 neurons in a larger cluster of 5. Rainer Gillespie, so Jan Jing, who many of you know from Aplysia work, he worked with Rainer Gillette before that, and he identified the AS1 to 3 cells that are 3 neurons that are in a cluster of 5, and they have contralaterally projecting axons. So they are homologs of the DSIs. And we base the homology by the fact that these neurons, again, are individually identifiable from animal to animal to animal within a species, but the same criteria can identify those neurons in another species. So again, serotonin neurons, 3 of them in a cluster of 5, contralaterally projecting axon. The only cells in the brain that are like that. And C2 similarly is a white cell that's located in a particular part of the brain, has a contralaterally projecting axon, and it's immunoreactive to fumaraphimid and SCP. In Tritonia, this labels 1 cell, and on each side of the brain. And the same thing's true in Pleurobenchia. 1 cell has those criteria, so this is a homologous neuron. When we look at the swim motor patterns, again, this is work from Jing and Gillette. The DSI homologs, the AS cells, and the C2 homolog A1 are both rhythmically active during the swim motor pattern. Now, they've identified additional neurons that in 40 years of research have never been found in Tritonia, and we've identified the VSIs, which they haven't found in Pleurobenchia. These are big brains, and so you can imagine you just didn't bump into that neuron, we haven't found it, or it could be that they don't play a role in swimming, and so we've never paid attention to that neuron. We certainly have never found a neuron that's electrically coupled to C2 that has the strong effects that A3 has. You could see its effects just by stimulating C2 because it feeds back inhibition onto A1 using Pleurobenchia nomenclature. And we've never seen that. So it's likely that either the cell doesn't exist or the synaptic connectivity is different. But they're playing similar roles. So they're both bursting during the swim motor pattern. Notice, and again, this is a subtlety that stomatogastric people might appreciate, but that the burst shapes are different. So it could be that the specific mechanisms of bursting are different. Now, Josh Lilvis looked at the serotonin modulation of the cell. So here's serotonin cells, here's C2, and he showed that in fact, here in Tritonia, we saw the C2 synapses are increased by DSI stimulation, and in Pleurobenchia, the same thing is true. The homologous neurons are also increasing the strength of so DSI causes C2 synaptic strength to increase. I'm using the Tritonia nomenclature here to avoid confusion. And serotonin does the same thing as well. So
Paul,
yeah.
Sorry, can I interrupt you for a second? So I don't know if you've seen it. Maggie asked on the chat
No, I haven't
about something 2-3 slides back. She was wondering if the recovery you showed before was affected or dependent on the synapse strength.
No. The initial susceptibility was, but then there was no further dependence on that. could some of them that weren't that susceptible, well, actually, I mean, you had to show susceptibility to show recovery. So if you weren't impaired, it's easier to recover. But we didn't notice anything related. It looked like it was a completely different, like recovery was a different phenomenon than sensitivity.
Thank
Sure.
you.
Okay. All right, so going back now to Tritonia and Pluribranchia, they both have the serotonergic neuromodulation, but maybe it's found in all of these animals and it has nothing to do with swimming. It's just like that's the basal condition. So Josh looked at he looked at Hermesenda then, which is a kind of a left-right swimmer. It just basically thrashes a little, but it doesn't swim like Tritonia and Pluribranchia do. And he identified the DSI homologs and he identified the C2 homolog, and it's immunoreactive to SCP and formurfamid. But DSI and C2 do not biorhythmic bursts of action potentials because this animal doesn't swim like that. And when he looked at the effect of DSI on C2 synaptic strength, it had no effect. He looked at the effect of serotonin and there was no effect. So Tritonia and Pluribranchia, so Hermesenda is missing the modulation that Tritonia and Pluribranchia both have. This suggests that there's a difference in the serotonin receptor expression in C2. So my student Ariana Tempakakis then went after that. So Adriano Senatori was a postdoc in my lab, and he initiated our work in transcriptomics. So he did full brain transcriptomes from several species, including Hermesenda, Tritonia, and Pluribranchia. And Ariana pulled out the sequences for 7 different serotonin receptor subtypes. She then looked to see which ones were expressed in C2 in Tritonia and Hermesenda. So this was quantitative PCR where she plucked out C2 from many animals and then tested individually for 5-HT1A, 1B, 2A, 2B, 4, 6, and 7, and found that the Tritonia C2 did not express the 5-HT4 or 5-HT6 receptor subtypes, but it strongly expressed the 2A and the 7. So then she did the same thing with Hermesenda. She plucked out C2 from several animals and then ran all of these quantitative PCRs, which is not an easy feat, and found that it expresses the 4 and the 6, but not the 7 or the 2A. So we found 2 different expression patterns in an identified cell in these 2 species. These receptors are found in the brain. So it's not that they're lacking the receptor, they're just lacking its expression in this one cell, although we didn't test other cells. So then what about Pluribranchia? So she showed, again, plucking out C2 homologs, that there's a 5-HT7 and a 5-HT2A, and then she decided she wanted to graduate, and she didn't want to get the negative results to do the others. So we switched, instead of doing single cell quantitative PCR, which is very laborious, she did single cell transcriptomes. So again, she plucked out the neurons and then sequenced them using, Illumina sequencing. And what she found, this is just a review of the quantitative PCR. So in Tritonia, you have the 5-HT1A, 1B, 2A, and 7, but not, and 2B and 7, but not the 4 and the 6. And the single cell transcriptomes completely back that up. These are the 2 that we think are important for swimming. In Hermesenda, it's missing those receptors, and the single cell transcriptomes completely agreed with that. Then in Pluribranchia, we didn't know about these because she only did the 2A and the 7 with the quantitative PCR. And so when she did the single cell transcriptomes, it completely agreed with those. We found that there was a 1B, but no 1A, 2B, or 4 and 6. So that suggests, again, that these 2 receptor subtypes were important for the modulation and important for the swimming behavior. So Tritonia and Pluribranchia independently evolved the same behavior using homologous neurons and orthologous 5-HT receptors, and Hermesenda has the homologous neurons, but lacks the behavior, lacks the modulation, and differs in the expression of the 5-HT receptors. But then Josh found something else, and this was a pain in the ass for him on his thesis work. If he looked at different animals on different days, sometimes they swam and sometimes they didn't. Ron actually had a term for this. It was the law of persnicketiness or something like that, right? It goes along with the law of beginner's
Internal
luck.
cussedness.
What was it?
The law of internal cussedness.
Internal cussedness. So Josh had a hell of a time because he would walk into the tank room and test the animal before starting the experiment. And a good portion of the times they just refused to swim. We couldn't figure out what it was. We really could not figure out what it was. We tried food, we tried light time of day, we tried all sorts of stuff, and they just decided one day they're going to swim and one day not. And these were all on the same day. So this animal swam strongly on day 2. This animal didn't swim very, many cycles. They were all over the place. We just couldn't figure out what the rhyme or reason was. But then Josh went ahead and did the experiment on the days that they didn't, if you clicked down an animal and it wasn't swimming that day, it didn't have the DSI modulation of C2 synaptic strength.
There is one more question from Astrid, and she asked, did the PCR and transcriptome also match in abundance, that is beyond presence or absence?
Yeah, so that's a good question, but we didn't have enough depth to really make that statement. We needed to get a larger sample size to really be able to know how many transcripts per million there were. It was just detection or not detection. Okay.
Good.
But what Josh found was that in animals that didn't swim, there was no serotonin modulation. And it wasn't because the DSIs weren't working, it was because the cell wasn't responsive to the synapse wasn't responsive to serotonin. And that there was this rough correlation to the number of swim cycles. So there's a bunch of zeros down here where there's no modulation and there's no swimming. So there was this correlation going on between the two. So then when Ariana looked at the quantitative PCR and she separated out the swimmers and the non-swimmers were not expressing the 5-HT2A or the 5-HT7 receptors in C2. And then when she did the quantitative PCR, the swimmers I showed you had it and the non-swimmers didn't have it. But the sample sizes are really low, so we were really worried about this. But none of the other receptors that she found correlated with swimming. There were differences across species, across individuals, but nothing correlated with swimming. And the lack of all of these not founds in pluribranchia is indicative of the low sample size. So I showed you that homologous neurons play the same role in species exhibiting the same behavior. I next want to talk about homologous neurons in species exhibiting different behaviors. So here's Melody leonina. It doesn't swim with dorsal body flexions, it swims with left-right body flexions, as indicated by the LR. This is thesis work of Jim Newcomb, who is now a professor at New England College in Henniker, New Hampshire. He identified the homolog of DSI in Melody. So again, in Tritonia, it's a serotonergic neuron in a cluster that has contralaterally projecting axon. Here's that cluster of serotonergic neurons. He filled it with dye. It has a contralaterally projecting axon. So it's a cell, it's a set of 3 cells that have the same properties from animal to animal and across species. So we call that homologous. He also identified, and this was his master's thesis work with Wynne Watson. He identified, or Wynne had identified previously, but a neuron called swim interneuron 1 that plays a role in swimming in Melody. So it is near the DSI homolog, so it's not serotonergic. It is fumarfimid immunoreactive, but it,
yeah, sorry to interrupt, but here is one more question from Ron Harrisvoric. He asks that, is it a feature of being erased in the lab?
We don't erase our animals, but I get it raised in the lab.
Right.
Autocorrect.
Basically, if the animals can't swim some days, they're not going to live very long in the wild.
Yeah, these animals, number one, we don't know why they swim. I mean, yes, Tritonia escapes from starfish. It's a really rare event. Pluribranchia is a nasty bugger. Nothing really eats it. It secretes hydrochloric acid. It'll swim in response to being bitten by another Pluribranchia, but they haven't been in our lab very long. They get caught and they get used within a couple of weeks. There's some of the old timers in the lab are there for a month or two. but they're pretty freshly caught from the wild. So this is, I think this is natural variability, and we thought about, you know, is the non-swimming a result of them being in the lab longer or not? So we looked at that, but it didn't correlate. That answer? Okay. All right, so Jim and Wynne Watson had identified, and also Stuart Thompson was an author on that first Melleby paper looking at the swim motor pattern in Melleby, that there's this neuron called SI1 that's near the DSIs and has an ipsilaterally projecting axon. And in Tritonia, the DSIs are rhythmically active, I showed you that, but in Melleby, their homologs are not. So here's the swim motor pattern in Melleby as monitored in swim interneuron 1, but the DSIs are just firing along as if there was nothing going on. In Tritonia, if you cause a DSI to fire, you initiate a swim motor pattern. So the DSIs, by releasing serotonin, cause the swim motor pattern. In Melleby, you can depolarize the DSI homolog and initiate a swim motor pattern. But then once you stop depolarizing, it goes on without it. Whereas in Tritonia, once you stop, the swim motor pattern stops. So the DSIs, again, are intrinsic to the circuit. The DSIs in Melleby are extrinsic to the circuit and having this modulatory action. So these homologous neurons are playing different roles in species with different behaviors, which you might expect that, you know, if you've evolved different species, different types of behaviors, and you have ancestral set of neurons, those neurons are going to take on different functions as the different behaviors evolve. What about these two species that have the same behavior? So Dendronotus and Melleby both have the same swimming behavior. And here's, I don't know, how is the video look? I want to make the video settings. Oh, it doesn't.
It's good.
It's not jumping. Okay, so anyway, they both swim with these left-right body flexions. And Dendronotus and Melleby belong to a clade of species that all swim with left-right body flexions. And this is an incredibly bizarre-looking menagerie of animals. These guys, Filaria, are actually pelagic all the time. They swim like fish. They do left-right body flexion, but they have forward propulsion with it. Others, so Melleby is also this is Tethys, which is bottom dwelling, and it swims just like Melleby. This is Melleby colmani, and it is this gorgeous animal. It's see-through except for all these, I don't know what they are, if they're muscles or brachial tubes or something. But all of these animals swim with left-right body flexion. So that suggests that it was present in the most recent common ancestor of the whole clade, which means that the behavior in Dendronotus and Melleby is the same behavior. It's homologous. So Kira looked at the swim circuitry. So here's the SI1 pair in Melleby, and if you inject current into one, it hyperpolarizes the other, so it's an inhibitory synapse. In Dendronotus, you inject current into one, it depolarizes the other, but it's not chemical synapse, it's an electrical connection, it's the gap junction. Akira and Jim identified other neurons that are found. Again, you can identify them based on morphology and other criteria to say that this is the same neuron. And the wiring diagrams are really different. So these two species both swim with left-right body flexions. They have homologous neurons, but those neurons are wired up in very different ways. So here there's inhibition, here there's excitation, here there's strong electrical coupling, here there's weak electrical coupling, here there's strong electrical coupling, here there's weak electrical coupling. Inhibition across the midline, no inhibition across the midline. And when you record from all of the neurons, so this is again like schematic Akira recorded from all 6 neurons at the same time, you can see that there's similarities and differences. So in Melleby, you get the left-right alternation of the swim interneuron 1s, but they're absent in Dendronotus because there's no inhibition across the midline. But you have the 2s and the 3s alternating, and in Dendronotus, you get the 2s and the 3s alternating as well. But those of you who are looking at phase differences notice that the left SI3 follows the right SI2 in Melleby, but it precedes the right SI2 in Dendronotus. And this is critically important for how these work. So in Melleby, I'm going to just give you the punchline, and I'll show you the evidence for the punchline afterwards. In Melleby, the 1s and the 2s form the oscillator, and the 3s act as a regulator to control the periodicity. So the 1s and the 2s burst on their own, through mutual inhibition across the midline. And then they have feed-forward excitation onto the contralateral 3, and then feedback inhibition onto the 1s and the 2s. In dendronotus, the 3 excites the 2 and initiates the burst. Then there's electrical coupling, and this becomes a positive feedback loop with its own intrinsic inhibition to limit the period and then cause it to switch to the other side. And we worked with Andrei Shilnikov at Georgia State University to model these, and both of these can be replicated with models for what it's worth. But we can also go do this with dynamic clamp. So first off, let me show you that the SI1, the 3-to-1 synapse, which is the same as the 3-to-2 synapse, is inhibitory in Melleby, but the 3s make excitatory connections onto the 2s in dendronotus. But both of these synapses are blocked by curare. So we think that they're both cholinergic, so that the 3s are cholinergic and they're making synapses onto the 2s that are through different nicotinic acetylcholine receptor subtypes. So nudibranchs have 24 different nicotinic acetylcholine receptor subtype genes that we found in the transcriptome. This was work of a master's student of mine that we actually haven't published yet. So some of them are anion selective and some of them are cation selective based on work that's been done in Linnea. So you can have both excitatory and inhibitory, but you all know that with regard to the motor neurons in the stomatogastric ganglion that can be excitatory at the muscle and inhibitory in the ganglion that are cholinergic. But if you put on curare, you basically eliminate the SI3 active synapses onto the other cells. And as I said, it was the regulator. So here it's bursting left, right, and now it slows down and becomes irregular because the phase switch is not there anymore. And so you're waiting for one of them to wear down before the other one can switch over and take over. So Akira used the dynamic clamp again and replaced the synapses that were blocked with curare. So now he put back the mutual inhibition among the 3s, and he put back the inhibition from the 3 to the contralateral 2. And didn't have enough electrodes to do all 6 cells, but this worked anyway. And when he flipped the switch, it starts bursting immediately. So all he did was replace the connections that were blocked with curare, and it sped up the motor pattern. In dendronotus, putting on curare, removing the 3s stops the motor pattern. There's no more bursting at all because there's nothing to initiate the next cycle. So the 3s were initiating the burst in the 2s, but if you block the synapse, then there's nothing, and it just falls silent or tonically. Again, Akira put in the synapses that were blocked, and it reinitiated bursting just like that. So in Melleby, there's continued bursting in curare, but in dendronotus there's not. And the difference is this part of the circuit where there's mutual inhibition across the midline and strong electrical coupling ipsilaterally. So what Akira did was he then rewired dendronotus to be like Melleby. It had the circuit topology of the other species. And here the SI1s and SI2s, remember the SI1s never fire and burst because there's normally no mutual inhibition across the midline. But when he flipped the switch on this one, it starts to burst. So this circuit topology is sufficient to allow these cells, the homologous neurons, to fire in bursts. So the homologous neurons are wired differently to produce homologous behaviors. The next question was, what the heck are the SI1s doing? And in Melleby, if you hyperpolarize the SI1s, they're part of the CPG. So if you hyperpolarize them, you prevent them from bursting, the whole thing stops. In dendronotus, if you hyperpolarize the SI1, you get maybe a little bit of slowdown, but nothing much. In Melleby, if you depolarize an SI1, you also stop because you hang it up on the depolarizing phase and it can't burst, and so it stops. But in dendronotus, if you depolarize it, you speed it up a little bit. If you have a prep that's not swimming, you depolarize an SI1 and it turns on rhythmic bursting. And again, it lasts beyond when the cell is active. And if you do a nerve stimulation to initiate bursting, you see that the SI1s are coactive with the bursting pattern. And when they stop firing, the system peters out. So Akira looked at the synapse from 3 to 2, and because he knew it was important. And he saw that before the swim, that synapse was weak. And during the swim, when the SI1s are firing tonically, that synapse is stronger. So he then did it a controlled situation where he just stimulated SI3 every so often, and then stimulated SI1 here and got a prolonged modulatory effect. So the SI1s are modulating the strength of the connection from SI3 to SI2. And it's probably using a neuropeptide for this, but we don't know. So then Akira did an artificial neuromodulation experiment. So instead of activating the SI1s, he just directly increased the strength of the synapse from the 3 to the 2 with the dynamic clamp. So here's the artificial synaptic current, and that's causing, initially the synapse is small, and the postsynaptic response is variable, but after initiating that, the postsynaptic response gets locked on. And when he turned on the dynamic clamp, he took a prep that wasn't oscillating to create an oscillation. Then he did a virtual antagonist, the opposite effect of weakening that synapse in a prep that was already oscillating, and that stopped the oscillation. So the strength of that synapse was necessary and sufficient for the oscillation. So let's bring this back to evolution then. So what I've said to you is that different levels of biological organization have separate evolutionary histories. The neurons are homologous, the behaviors are homologous, but the circuitry between them has diverged. Okay. This is where I got a lot of pushback when I talked about this at Cornell, because a lot of neuroscientists say, well, if it's not the same circuit, it's not the same behavior. But what I'm saying is that there's different levels of biological organization, and you can have the behavior be conserved while the underlying mechanism changes. And so I like to show this picture of the frogs, because I think everybody on the Zoom call will agree that the left front leg of this frog is homologous to the left front leg of that frog. But the developmental mechanism that gets you there is diverged. So in this species, the left front leg emerged from the wall of a tadpole. And in this species, the left front leg emerged from an embryo. So the history, the way that you get the behavior has changed, but the behavior is still the same. The way that you get the leg has changed, but the leg is still homologous. All right, so I showed you Melody and dendrinotis different conductivity and the activity of SI1s. So a student of mine, Chiruni Gunaratne, looked at Flavolina, another species that swims with left-right body flexions. And she found a third circuit motif. So this species swims with left-right body flexions, but there's not strong electrical coupling across the midline. There is indirect inhibition across the midline. And the SI1s then are rhythmically active, just like they are in Melody, but a different circuit motif for Melody. Then she looked in Tritonia. And in Tritonia, there's strong electrical coupling like in dendrinotis and mutual inhibition like in Melody, but Tritonia doesn't swim with left-right body flexion, so during the dorsal ventral swimming, they're actually inhibited. So there's 4 different circuit motifs, and the homologous neurons exhibit different connectivity each time, and that's not reflected in the phylogeny or the behavior. So it suggests that if you want to use connectivity as a way of, well, if it's true in this species, it's going to be true in this species. It seems like connectivity is the least conserved feature of behavior or of cell identity. So the identified neuron approach is powerful, but it's limited. We can only find the cells that we can find, and we're poking around blindly in a brain of 5,000 to 10,000 neurons. So I wanted to look at another species and maybe one that doesn't swim and see what these neurons are doing in that. And this started as work of an undergraduate in my lab at Georgia State. So Amira, on her own, figured out that we could raise this species. She tried raising some of the others in the lab and it didn't work. But this species, as it turns out, is commercially available. Oh, and by the way, she applied to Illinois State for grad school, just saying. So the Bergia goes from egg to adult and back again in less than 2 months. So now we can get lots of animals. We can do developmental things. We can do all sorts of observations that we didn't have. And it's much more accessible for undergraduates to work on because the animals aren't that valuable. We can get them easier than you can get crabs. So this allowed us to get a Brain UO1 award and begin a collaboration back with Bill Frost. So I'm working with Bill again after 20 some odd years. Jeff Lichtman at Harvard, Dee Dee Lyons at Scripps, and Vince Lejinsky at the University of Maryland. So the idea is to get a complete brain atlas, describe the behaviors, record, because these guys don't swim, like what do they do? Record from the neurons during the behavior, develop transgenic nudibranchs, and then model these networks in activity. And this has now led to a U19 proposal with 13 institutions and an expanded list of collaborators. So this has been a real boon to what I'm doing. And as Ron said, it's completely changed how I think about these animals. So again, the goal is to get a complete brain atlas, to do EM, reconstruct it, segment it, and then get a connectome. At the same time, identify the neurons and then create understanding of what those neurons are through gene expression differences and neurotransmitter and so forth. So again, we can identify neurons based on immunohistochemistry. Different neurons express different types of receptors, different types of transmitters, but we wanted to know the ones we didn't know. So T. Bui was a master student in my lab. Des Ramirez is a postdoc, and they dissociated the neurons and then did single neuron sequencing using 10x genomics, did a cluster analysis. So this is just the first attempt, but we're able to cluster different neurons. And each of these dots represents a different neuron in a space that represents the differences in gene expression. And then this is another way to represent those 24 clusters, showing some of the most highly expressed genes. And what you can see is, for example, that cluster 6 expresses these particular genes more than the cluster 7 does, and cluster 9 and 10 express these genes. So we can begin to then find the genes that differentiate the types of neurons and then map them back out onto the brain. So this is using a technique called in situ hybridization chain reaction, which is a more sensitive way of doing in situ hybridization, and it's fluorescent, so we can multiplex it. So the blue cells are expressing the neuropeptide APGW amide, the yellow cells are serotonergic, the red cells are cholinergic. And there's a lot of cholinergic cells. And then we're working with Jeff Lichtman's lab to do the full brain connectome. This was started by Brandon Drescher, who was a postdoc in my lab, who actually moved to Cambridge to spend time on Jeff's machines. This is an automated ultramicrotome where you do the sectioning and the sections float out onto a tape that's then put out onto a silicon wafer and then imaged in an electron microscope. And we started to do this and then COVID hit. We got there. So this is just to show you, this is the face of the Bergia. This is its rhinophore, which is the olfactory organ. Here's its eye, its oral tentacle. The brain sits between there. This is a clarity cleared animal. So this is the position of the brain in the animal. This is the rhinophore nerve that goes up into the rhinophore. This is the rhinophore ganglion, and which I thought naively was like a antennal ganglion or antennal lobe in insects, or like the olfactory bulb, or an antennal lobe in crustacea too, but it's not, as it turns out. And then this is the rhinophore connective, which is the connection between the rhinophore ganglion and the cerebral ganglion. So again, we started at the front and got stopped right there, and we got this image. And this image changed how I think about this brain. It profoundly affected the way that I thought about it. This is a cross section of the rhinophore connective that shows the axon profiles. And if we just take a zoom in view here, this is 1 micron. Some of these axons are 10 to 50 nanometers in diameter. And there's all sorts of structure in here. So there's glial cells. If you look over here, you can see differences in ultrastructure of different regions. There's dividing lines, there's large cells, there's small cells. And when Yaron Mayerovich, who's a postdoc in Jeff's lab, looked at this, he recognized that the ultrastructure looked pretty similar to octopus. And he has been working on the connectome for the octopus vertical lobe. And he applied the ground truth from the octopus to Bergia and was able to automatically segment this nerve, the connective, and counted that there were 30,000 axons in this connective. So this totally changed the way that I was thinking, because you have a brain of 5,000 neurons getting 30,000 from each rhinophore. This is the partially segmented or partially assembled rhinophore ganglion. So this is a high resolution image going through, and you can see the cell bodies in the nerve hill. And this is now allowing us this is where the video freezes. Okay, so this is where it's allowing us to now segment out the neurons from that volume. So we're going to do this with all the neurons. I've got a class of undergraduates, I should say, a chain gang of I don't know, what do you call a group of undergraduates that do forced labor like this? Something like that.
A murder of undergraduates.
No, only crows. Cheyenne Tate is another postdoc in my lab, and so she was curious about where those 30,000 axons went in the brain. And so she did a neurobiotin backfill of the rhinophore nerves, and she got this image showing that this is, if we had seen this before we saw that EM, we thought this is just junk. Look at that, there's just a smear, but that's 30,000 axons filled with neurobiotin, or a subset of 30,000. And then many of them are going to the contralateral rhinophore ganglion. And then there's particular regions in the brain where they're going. There's particular neurons that backfill. But the fact that they're going contralaterally, many of them going contralaterally, actually answered a question that Phoenix Quinlan, who was a grad student in my lab, had. She was doing a Y maze, or T maze in this case, putting the prey item in one arm, and the animal would get to that arm almost 100% of the time. But if she took off one rhinophore, they couldn't do the task anymore. And of course, if she took off both rhinophores, then they went to chance as well. So they need to make a comparison between the two rhinophores, and this is probably the biological basis for that comparison, that they're going, the neurons from the rhinophores are going directly over to the other side. The other thing that Cheyenne found was this. This is the eye. That's a photoreceptor that backfilled by putting tracer in the nose. Okay, so we put the neurobiotin in the rhinophore and filled a photoreceptor. This wasn't expected, but maybe it should have been, because in 1978, Dan Alcon published this paper that showed recordings of photoreceptors, and if you put on an odorant, it hyperpolarized. So this is suggesting that there's really early connections between the primary senses. And in fact, the whole Hermesenda learning field is about the status cells, the hair cells, the learning is occurring in the photoreceptor. So early on in processing, there's interactions of the senses. And so you really can't talk about the primary senses as different pathways. So when she filled the actual primary sensory neurons, she found that many of them that synapse within the rhinophore ganglion, but a good chunk of them go on into the brain and make synapses all over the brain. So there is no olfactory area of the brain. The brain is an olfactory structure. So this has got us thinking about the octopus again, because the octopus has this embodied intelligence. It has 170 million neurons in its brain, but 330 in its arms. This is a patch of Bergius skin with the serrata, and you can see that it makes directed movements with no brain there. So there's this peripheral plexus of neurons that we're examining now to see how that is controlled and interacts with the brain. So Cheyenne did the next experiment where she literally just dissected the brain out of an animal. And so this is a brainless slug, and this is the slug with the brain. And if the movie decides it's going to play, you'll get to see that. There we go. Come on. So this is an animal without a brain. It's much slower. This is sped up 5 times. This is normal speed. You can see that they use their oral tentacles for exploring the environment. The oral tentacles are moving just fine. Well, not just fine. They're moving. And the animal actually will turn. It will avoid obstacles, but it doesn't have a brain. So that was it. That was my journey from GPR to SI1. And these are the people who did the work. So I talked about them in the parts of the seminar. Our funding from NSF and from NIH. This is what we look like just pre-pandemic, and this is what we looked like a week later. So thank you all very much. Wait, what do you mean surprisingly? They're notoriously cute.
Very nice.
Sorry?
Thanks, Paul, for this talk. So it's now open for discussion. If you want to ask any questions, please go ahead.
Yeah, I'll ask a question. So I'm trying to put it all together, Paul. So you found a lot of things that are conserved and a lot of things that diverge during evolution. And I'm trying to figure out order and importance. So in your early work, say with the GPRs, the big story was the same neurons are there, they look pretty much the same, and they vary which transmitters they release. And then you've shown an enormous amount of work.
More than that,
Yeah.
because
Right.
there were also 4 cells in some species, 2 cells in others. So you have duplication.
And
You
there's
have
also-
segmentation into separate muscles versus one muscle.
Right, right. I'm just trying to come up with big categories of things that change. And
Okay.
the transmitter release can vary. The synapses can be added, can be subtracted, can become more chemical or electrical, can change the receptors so they can turn upside down for the same transmitter in the same neuron, the same postsynaptic cell. And obviously, homologous neurons can remain homologous or drift apart, or new neurons can show up in a new species that appear not to be present in the other species. So these are kind of big classes of the big changes that can occur. Would you rank those in terms of the critical issues for evolution of behavior between species?
I think you have to consider scale for one thing. You know, if you're looking across 100 million years of evolution, or you're looking across 50 million years of evolution, that matters a lot. Tritonia and pluribranchia are separated by over 150 million years. So that means, I mean, mouse and humans are separated by 65 million years, just as a comparison. So they're more distantly related to each other at time than mice and humans are. But they have the same neurons. They don't have, there are different neurons. There's no question. We're seeing, and this is the stuff that we're seeing now when we really examine like all of the neurons in the brain, there is variability, and there's variability even within a species. So those types of generative processes are going to determine how conserved something is. So a lot of it gets back down to development, which I know you hate. But I've
changed my mind on that, by the way.
Okay, good. So we're looking at, that's one of the things that I wanted to do. That's my motivation for going to Bergia, was so that I can say, well, actually, I wanted to do this with Melody and Dendronotus. When does the SI1 diverge? Like, when does it start expressing these receptors versus these receptors? Because if that's all it is, that's pretty easy how you can imagine that to happen. So it's turning out to not be such a simple question. Nobody knows how you get these neurons to be identified. In other words, where do identified neurons come from? We know this in some with C. elegans. We know this with insects to some extent. We don't know it in mollusks. We don't know if like they're determined from birth, or they're determined by cell-cell interactions. Is there a proliferation? And like the next one in line is going to be it. In the stomatogastric ganglion, there was some work for a while in what determines it. And Dirk can correct me if I'm wrong, but my understanding is that some of the earliest neurons are the interneurons, and that the motor neurons are appear later. I don't know if you're the right one to ask, but I think the PY neurons and the GM neurons are born last, and they are the most variable.
Well, it seems a little bit, and it's not. There's no real simple way to show this rigorously, but there's as if there's a cluster of a certain number of cells, and then there's individual difference in how many of those become GM neurons and how many become PY neurons, which is weird because they don't even have the same transmitter phenotype.
But if they are born later, then that could be why there's fewer restrictions on them. And
yeah, they're also among those that are more than 2 copies, right?
Right. So right. So how do you always get 2 PDs? And the difference is, this was actually something that I've been thinking about since the early days too in stomatogastric, that the differences in the electrical coupling in crab versus spiny lobster, but crab and spiny lobster are really separated. In fact, I don't know what the latest is on the phylogeny, but spiny lobsters are often their own when it comes to the other decapod crustacea. And I don't know if has that been resolved molecularly? Does anybody know where they come off from, shrimps versus crabs? Crabs are this amazing explosion, diversification once the brachyurans. So looking across crabs could tell you something, but then once you start to look across these wide swaths, it's really kind of hard. But it's amazing still the extent of conservation of cell types. The fact that you could find A, B, P, D, P, Y in Pennularis and in crab and in shrimp. So that's incredibly conserved. But the things that you see differing are the details of the synaptic connectivity, just like we do.
But actually, Paul, there is another difference that I wanted to ask you about, which is, in your case, you've shown all these differences in sort of circuit diagram, and I'm not even talking about the dynamics of synaptic function and intrinsic properties. And you basically, at least in Josh's work, it seems like transmitter phenotype is conserved enough that you can actually use it for determining the homologs across different species. Whereas from what little is known in the STG, the circuit doesn't change much. I mean, the dynamics changes, but the circuit diagram has like very minute differences. We have the same things, just anecdotally, that you have seen that the neuromodulator receptor complement can change all the way to, in one species you have a D1 receptor, or in the other species you have a D2 receptor and things like that. What does change, though, what it seems to be more malleable is, from the few examples that we have, is the modulator phenotype itself, particularly for peptide co-modulators, that homologous neurons across different species just have a completely different subset, which is completely different from what you've shown, right?
Not quite. I agree with you. I agree with everything you said, but I think that, and I think this is a really important point that gets to what Ron was asking. First of all, GPR cells actually differ in transmitter phenotype because in Pennularis, they're not serotonergic. And that's probably because the GPR is using a borrowed transmitter. Serotonin is probably not being synthesized in the GPRs. It's only being taken up and used. So all you have to do is get the one gene for the transporter, and now you've got you change your phenotype. I don't know, there's probably other genes you have to do as well. With regard to the peptides, I don't know yet whether certain peptides, yes, seem to be diagnostic of the cell type, but we are seeing large differences in how many cells of a particular peptide phenotype are present, and we don't know if those are differences in the numbers of cells or in the expression of that gene. So we're looking at that. One of the things, this has actually led Cheyenne to study sex. Do you want to see some slug sex pictures?
I'm not going to answer that.
Let me just quickly share this. This is another story. I wasn't going to get into it, but since you asked. Here's some nudibranchs that are having sex. They're about to have sex. So they encounter each other, and the first thing they do is they line up their mouths. So I have to tell you all, they're hermaphrodites. So they're both male and female. Here they're lining their mouths up, so they're kissing. This is the first step of slug sex, is to kiss. And then they're going to mutually exchange spermatophores. So you take my sperm and make eggs, and I'll take your sperm and make eggs. And you can actually see it happening. It's going to happen in a couple of seconds. I don't know if that is as interesting to you as it is to me to see that, but the thing is that they lay eggs. I'm just going to go to the next slide. They kiss, they transfer sperm, they cuddle. There's another important aspect. They cuddle for hours after they have sex, and the reason they do that is because they're preventing the other one from reaching back and eating the sperm, because they don't want them to cheat. The way you cheat if you're a hermaphrodite is you don't carry out your part of the bargain of raising the other one's eggs. You just act as a male. And here are the egg spirals. And so Cheyenne actually found the egg-laying hormone. So we have and synthesized it, injected into animals. It causes them to squirt out eggs. And then she looked for the expression of these. This is where I'm getting to the peptides. So she looked at the expression in the brain. And so there's a scattering of cells that express the neuropeptide. And then you see differences across individuals and how many are expressing egg-laying hormone. And then she noticed, oh, these guys don't have gonads, and these have gonads. These are probably homologous to the bag cells of Aplysia. but these are not developmentally different from each other. These animals all came from the same, they're all siblings. They came out of the same egg strand, and they have differential growth rates. And she thinks that these are actually an alternate mating strategy. So if they're in competition for food, some of them just don't get big and don't compete for the food, and they don't make eggs, and so they cheat by having matings only as males. And that's reflected in differences in their brains. So I don't know if there's cells that are here that are not expressing the peptide, or if they make new cells. And I don't know if it's reversible or not. So this was a long-winded answer to say that there's still a lot we don't know about transmitter phenotype and neural identity. I don't know if that answered your question or if I went off on a tangent. I just wanted to show you that data because it's really cool.
I know. I don't think that probably isn't a good answer, but it is an interesting question if like changing either the circuits themselves or the transmitter phenotypes actually, it's sort of like a different functional thing to like a different strategy to you know, change behavioral basically, or if that's just sort of random, whatever you started with.
It also, well, I don't know about random, whatever you started with, but it also depends on the promiscuity of the receptors. I just said sex and promiscuity and cheating in the same talk. But the receptors, if the receptors have high fidelity for just one neurotransmitter, then it doesn't do any good to change your neurotransmitter. You have to have postsynaptic receptors that are, that are matched up. And aren't the peptide receptors? So you talk about the receptors having the same actions on the same currents. Jorge talked about this last time, but are the receptors known to be different? Or are the peptides that are acting on the proctolin receptor, are they acting at different receptors and having the same effect on the same conductance? Or they're also different, or is it possible that this one receptor can be activated by different peptides?
Well, they're likely more than one receptors, but how promiscuous each individual type of receptor is, we don't know. But the suspicion is that they're not super selective, at least for different isoforms of the same sort of group of peptides.
Well, can I ask a quick question? Yeah, so when we say, why don't we see a given cell type that's responsible for a behavior in like Trittonia versus STG, like we're in the STG where we have, two identified PDs and you can generally identify them across preps pretty reliably. Is it a fair comparison to ask that, where we know that, STG is basically always on? So you have homeostatic rules that have been described by, Jorge and Dave Schultz and others about ongoing activity and modulatory related homeostasis. So is it, and we know that the swimming behavior isn't constantly on, right? In animals that have more neurons than STG crab, we know that there can be like representational drift. So a population of neurons could take over the role of a behavior and it can change in time, right? So do you think something like that is more likely to be the case when you have more neurons available to you instead of just like 30 some?
Yeah, I think that the stomatogastric ganglion is developmentally constrained in many respects. It's also functionally constrained. You have to have a working foregut and gastric mill. The animal can't, it's not an option to not have it. So the things that are evolving are going to be, there aren't that many, functionally, I don't know what's changed in the millions of years of evolution of the decapod crustacea with regard to the function of the foregut. There's differences in feeding, but across the Brachyura, even in crabs, you have massive differences. I think there was a paper that just came out on this. You have filter feeders, and you have animals that are capturing large prey items, and others that are sifting through small, but they all have a gastric mill and they have a pylorus. And I don't think that those circuits are very different. The teeth, this is in my thesis, and this is in a paper that's extinct in the literature now, in seminars in neurosciences, have tremendous variability. So the structure of the periphery is tremendously variable. The musculature varies a lot, but the circuits, and again, if you look just within Brachyura, just across the crabs, the circuits are similar. If you go from crabs to spiny lobsters, you do see big differences, and having to do with electrical coupling, the activity of the neurons, and so forth. But the modulatory differences is an interesting question, because if the modulatory differences can drive differences in the dynamics of the circuit, then that could get it, that you don't necessarily, there's no pressure to change the circuit, and it's very much safer to keep the circuit the same. But I think, Omar, your question also, if you've got a larger pool of neurons, you've got 10,000 neurons, if you double a couple of them, you got a few more to play with, and there's no cost to doing that. So you're not constrained as much in a teleological way. So the nervous system has fewer constraints if it's larger and less well defined than a small 30-cell ganglia.
Yes, thanks.
Any further questions? If not, I have one final question, which may be a little stupid, but I'll ask it anyway.
Wait, I want you to take that back right now. I will not answer it if you say that it may be stupid.
So this is about the homologous neurons and the networks that you've shown. So these connections that you've shown, do you know if there are any more connections that are just there anatomical but not functional, or is this?
It's an excellent question.
Did you just poke it with electrodes and see if there's something happening in the pre and post synaptic neuron, or do you have like already EM slices where you can see if there are anatomical connections between?
No,
these
that's why I
neurons?
want to do the whole connectome, is that we don't have that information, and we want to get that information. But that's not a stupid question. That's a really smart question. What we saw in Tritonia was that we thought, like DSIC2, that's been studied for decades, and then when you injure it and then you look later, when it recovers, then suddenly there's polysynaptic activity that wasn't there before. So the system is not static. And it could be use dependent, it could be, experience dependent, and there are individual differences. So most of the time, this is, Eve's good enough stuff, it works. So it doesn't have to be the same every single time, but that may be the substrate for further change in other directions. Does that answer your question?
Yeah. Sounds like there's still a lot of work to do.
Oh, yeah.
Right. Any other questions? If not, Farzan, Smita, Nelly, any final words from you?
I don't think you asked any hard questions.
Next talk. What is the next talk? That's what they're asking, Smita and Nelly.
Next talk, yes, next monthly meeting will be on Thursday, April the 15th. And I'm glad to announce that women are back, and Gina Turigiano will be presenting next meeting.